afterwards. So while I'm introducing our next speaker, Sheila, go ahead and you can put your slides up. So thank you so much, Margarita, for that fascinating talk. And now next up, we're going to hear from Shiladitya Banerjee. So Sheila is an assistant professor of physics at Carnegie Mellon, working in the area of theoretical and computational biophysics. He did his undergraduate studies in India. He earned his PhD in physics from Syracuse. He did a postdoc from the University of Chicago. He joined the faculty of the University College London. So he's been a lot of places. And current research in the Banerjee group at Carnegie Mellon is focused on understanding the role of geometry and mechanics in cellular decision-making and collective behavior. So with that, Sheila, take it away. Thanks. You're muted.
Hello to everyone and who is joining here today, and thanks to Ankita and Jen for continuing to run this seminar series. So my talk today will focus on the role of epithelial cell junctions on collective movement and morphogenesis. And I'm really here as spokesperson of the work done by my former PhD student, Mike Stadden, who recently moved on to a postdoc position at Max Planck CBG. So this will be an overview of some of our recent studies focusing on modeling, which is what we do, okay? So as you all know, in development, epithelial tissues undergo complex shape changes. Shown here is a movie of a very familiar movie of Drosophila germ band extension. And what you see is that as the tissue elongates, you see dramatic cell shape changes and rearrangements at the scale of individual cells, right? So while the tissue elongates, what you observe is that this elongation is really driven at the cellular level by intercalations. So cells intercalate. Like rows of cells intercalate to form fewer and longer rows, and this actually drives coordinated cell movements that can result in intercalation, right? And then what's really happening behind this intercalation at the subcellular level are these pulsatile activities of Rho and non-muscle myosin that drive this ratcheting like contraction that drive this elongation, okay? So what I have here is that I can construct a sort of feed-forward loop, right? Subcellular activity, let's say of Rho and actomyosin, that drive cellular level remodeling in terms of shape changes or rearrangements, and that can lead to collective movement in tissue morphogenesis, right? So this is a well-known example that we know of in tissue morphogenesis. And what I wanted to discuss today is our work on understanding this in the context of collective cell migration and motility, okay? And so my talk here today will have two parts. The first part will talk about how the local cell movements coordinate collective cell motion in the context of gap closure and wound healing. In the second part of my talk, I will briefly touch upon some of our recent work understanding what type of mechanochemical feedback circuits underlie cell rearrangements that can actually lead to global shape changes, right? And if we look at it, the traditional understanding is that we think about cell migration and movement in terms of activity, right? So Rho and RAC sort of drive motion and migration. But when we are talking about motion in the context of multicellular systems, you have to work against the mechanics of the surrounding tissue, right? So tissue solidity will sort of inhibit motion. So you need to overcome that stress, and the way to do that is through junctional remodeling of intercellular junction, and that is also driven by activity, subcellular activity, right? So few years ago, working with my former colleagues at UCL, Rob Tetley, who is I think in the audience today, and my former colleague Yanlan Mao. So we were studying wound healing in Drosophila wing disc, okay? And so this is Rob's experiment. So what he found was that the wound healing is coordinated in two phases, and in the first phase, that there is a rapid contraction of the wound, which is driven by assembly of actomyosin purse strings, shown here in magenta. So purse string assembles, it contracts the wound edge, and that drives motion. But the second half, in the second half of the closure process, what happens is that you see this rapid cell rearrangements that occur at the edge of the wound, and so basically cells actually intercalate away from the wound edge, right? And so we call that a fluidity phase. What's really happening is that the cells are rearranging and reducing the number of cells at the wound edge. Okay, so there are two phases. Initially, is driven by actomyosin tension at the purse string. Secondly, there is a phase of fluidity that is necessary to drive intercalations. So using modeling, we were looking at what is the value, like what is the utility for having these intercalations, and we were able to reproduce, wound closure in tissues without cell division, etc., just by having intercalation and purse string driven motion, right? Now in simulations, if we disable the intercalations, what happens is that the tissue gets really jammed and it's difficult to close because there's a huge buildup of stress, and then there's no way to relax those stresses, right? So so we need these intercalations to drive collective movement, right? And so we then looked at using simulations like how fast you can migrate, and then how does that depend on two factors. For example, one is the interfacial tension between cells, which actually regulate tissue fluidity. And the other is the contractility of the purse string that assembles around the wound edge, right? So as you expect that reducing interfacial tension, which will increase tissue fluidity, you actually close the wound faster. But you can also do so by increasing purse string contractility, right? And the wild type tissue somewhere sits in between here, marked by this black asterisk. And so what Rob and Yanlan did was to test this in experiments by using mutant flies, where in one case you inhibit myosin and you see that the closure is faster. And in one case where you actually overexpress myosin and you see that the tissue is too tense to close, okay? So so overall, this sort of tells us that tissue fluidity and is really helpful to have collective motion. So so in recent work, we saw more examples of this, and I'll particularly focus on one of our recent studies that published this year, where we're looking at neural tube closure in mouse embryos. Okay, and this is a really gigantic gap. This is not a wound, and this is a gap that forms in development about 500 microns long. And this is a work done with my former colleague at UCL, Gabe Galea and Erini, who is in the audience as well, I think. And so as you see that the gap closure in this mouse embryos occur in a very precise fashion, right? And you see an asymmetric gap, and that asymmetry is maintained throughout, right? The shape of the gap is also maintained throughout. And if you stand for actin, you see that along with the assembly of sort of this belts of actin, you also see lamellipodia-like structures that sort of drive the motion, right? So you see these ruffles that assemble at the edge of the gap, right? So if you sort of characterize the motion of the gap, you see that closure is happening faster from one of the ends and slower from the other end. And that is really necessary to maintain this asymmetry in the shape of the gap. And what was really striking is that unlike closure that is driven by actomyosin purse string, here the aspect ratio of the gap is maintained at a constant value, right? So and to sort of explain that, we developed a model. We used the Vertex model, but we needed to include an additional mechanism for closure, which is active cell movement, right? So cells crawl along with purse string driven contractility, because purse string driven contractility. So purse string acts as a cable under line tension. So what happens is that as the gap closes, the purse string will become round, and the asymmetry of the gap will be lost, right? So to maintain the shape of the gap, we need something like active cell migration, okay? And if we include this active cell movement along with purse string contraction, what we can recapitulate some of the experimental data that the asymmetry of the gap is maintained and the aspect ratio is maintained, right? But underlying all this, what seemed important for the gap to close was, again, intercalations, right? So if we count the number of cells surrounding the gap in the row 1, row 2, as well as in row 3, we see that there is a steady decrease in the number of cells around the gap. So cells rearrange and intercalate as the gap shortens, right? And this was really important because without this intercalation, what would happen is that there would be a huge buildup of stress at the edge of the gap, okay? And then you would need sort of more forces to sort of close the gap. So, and this is also something we found in the experimental data looking at intercalations. You can see that cells actively intercalate away from the cells, away from the gap edge, and over time the number of cells around the gap reduce. Okay, and if you did not have that, that would create some sort of defect in the patterning of the epithelia. Okay, because you will likely have more rosette-like structure, something that you don't want here. Okay, so so these are sort of two examples in vivo where intercalations play an important role in collective cell migration. We saw similar things working in vitro. So this was work again done with 2 years ago with my former colleague Mike Model, and what Mike was looking at is this in vitro MDCK cell layers on 2D substrates. And here actually, like in the previous case, closure was driven both by lamellipodial migration and actomycin purse string, right? So what happens here is that the cells initially starts to crawl and actively migrate, and then over a longer times, they sort of transition into a purse string type mechanism of closure, okay? Even in this case, we saw that intercalations played an important role, okay? And the more intercalations you had, the closure was faster, okay? And we could recapitulate all these results in our simulation, but one sort of result that I think is very interesting is that this intercalation-driven closure is very efficient when you have both these mechanisms. When you both have purse string-driven contractility that drives cell motion, as well as you have lamellipodial migration. So when you have both these mechanisms, active mechanisms of closure, intercalations are the most efficient, okay? And that's sort of detail in this paper done a couple of years ago. So what I told you so far are some examples on how intercalations and cell level rearrangements help in collective motion and collective movement, right? So that's all very good, and we could actually capture most of these results in sort of simple models of tissues, okay? By simple models, I mean that, using sort of canonical models where we describe tissues as a collection of mechanical objects like cells, and cells have tension, and then, they can intercalate, etc. So there was no surprise there. But so what we wanted to know is what is the underlying mechanics of this junctional remodeling, okay? And so there we found sort of evidence that the models that we think are good and they work very well at the collective scale, they don't work really, they fail miserably at the scale of junctions and individual cells, right? So the question we wanted to ask is that if we have a sort of like pulsatile activity, as we saw before, like in the pulsatile activity of Rho and myosin can drive junctional contraction, right? And that leads to morphogenesis. So if you simplify it and ask this question, like if there is a pulsatile activity, let's say of Rho and actomycin, and how does it lead to sort of plastic deformation at the scale of individual junction, right? So when a signal is on, you expect that the junction will contract. And when the signal is off, you expect, if the tissue behaves like an elastic system, you expect that the junction will return to its original length, but that doesn't happen, right? So there is some sort of plastic deformation that happens, and so we wanted to understand what is the underlying mechanics for that, right? So to do that, we worked together with Margaret Goodell and her former PhD student now, Kate Kavanagh, who built this optogenetics system. So basically, what this is, so we wanted to control spatiotemporally row activity, in a monolayer of Keiko cells, right? So this is an in vitro system, which is very stable cell line, and then the idea was that using optogenetic tools can spatially control row activity, so you can actually induce contractions in your in a desirable fashion. So you can design maybe the sort of time period and the amplitude of this contraction and see the mechanical response of this junction in response to it, right? So what Kate did, like, she used an optogenetic system on model epithelia where blue light activation causes recruitment of GEF on the membrane, and which can then sort of activate row locally. So here you see on bottom left a movie of junctional contraction. So by shining light, you see that the junction contracts. It sort of comes back, not actually. So what you find here is that if the light intensity is high enough, you see plastic deformation. So there is contraction over as long as the light is on, but once you turn off the signal, you see that the junction remains contracted, right? So using a low light intensity of rho, you see that the contraction is completely reversible, right? So it sort of depends on the strength of the applied light intensity, okay? So if I translate this into physical language, what it means is that if you apply a higher contractile force, you see a larger, like an irreversible contraction. But for a smaller amount of force, you see a completely reversible contraction, right? And this contraction saturates for longer time scales. So if you have the activation for longer and longer times, there is a finite limit to how much you can contract, right? So what we see is that with a single pulse of rho, you can only contract maybe 80% of the length, right? So then how can you actually go all the way to intercalate, right? And the answer was that you need to have pulsatile reactivity to enable junctional ratcheting, right? So inducing multiple periods of rho activity can actually contract the length all the way to intercalation, right? Which we actually didn't do here, but this sort of shows that the sort of value of this oscillatory ratchet mechanism of contraction. Okay, so this sort of response, like this mechanical response, is very novel. Okay, because it's not explained by existing models. And what I'm trying to say here is that if you use experimental existing models such as Vertex models, or any variants of it, like all the existing variants that we know of, so it comes in different forms. Vertex models have been around for some time now, and it comes in different forms. So the basic idea is that cells are made up of polygons. The polygons edges carry some sort of line tension. The cell has elasticity. And what was shown in the work by Max B and Lisa Manning and others is that you can tune the mechanical property of the Vertex model to behave like a solid or a fluid, okay? And if you look at a solid tissue, you see that after a local contraction, you get a complete reversible recoil of the junction length. But if it's a fluid-like tissue, there is no recoil, right? So you get a complete remodeling. Okay, so this didn't explain the behavior that we expect. So what we did is that we sort of built a new model based on the existing data, and there was a lot of trial and error, and so it took us some time to come to this model. But let me try to explain it in one or two slides. But please ask me questions if something is not clear. So what we found was needed was that to have permanent length changes like plastic deformation, you need tension remodeling. So this can be understood simply from this mechanical argument, like when you have contraction, like and you want to stabilize this contracted length, to balance this, you need to have a higher tension, okay? So as you're contracting, if the tension is also increasing, then you can stabilize that length, okay? So that was what was inside this model, like, if the applied strain is below a certain threshold, nothing happens. If the applied strain is above a certain threshold, you get this tension remodeling. So it's sort of like an excitable mechanical system, okay? And but tension remodeling is not enough to stabilize, because with continuous tension remodeling, the junction can just collapse onto itself, okay? So you need some way to dissipate the stress, and that was something we call strain remodeling. So there is continuous strain relaxation in the system to limit the amount of junctions shortening. So what happens is that as the tension is increasing, the strain is also relaxing. And as a result, so both these mechanisms can give rise to the expected behavior that we see in the experiment, that when the applied force is above a certain threshold, we see irreversible changes in junction length. But when the force is below a certain threshold, we see reversible behavior, okay? And underlying this, sorry, I think I skipped a slide. So underlying this novel mechanical response at the molecular level, what's happening is that this junctional remodeling was coordinated by endocytosis, okay? So the junctions would contract, the membrane would buckle, and then endocytosis would happen, and that would stabilize the length, okay? And this model can faithfully capture junctional ratcheting. Like with these two mechanisms, we can actually capture junctional ratcheting. So, one of the interesting results that came out of this is that what is important to get maximum contraction is not necessarily the amplitude of the activity, but the time period, okay? So what you see is a heat map of the amount of the final length of the junction over the initial length. And on the x-axis, we have activation time, like how long you activate the particular junction. And on the y-axis, we have rest time, like how long do you wait before you apply the next pause. So the longer you wait, it's actually better. Like you can actually maximize contraction by waiting longer and activating longer, right? Because if you wait longer before applying the second pulse, the system has enough time to sort of lose all mechanical memory. And then it can again contract, right? So you need to give the system some refractory phase where it can sort of forget its mechanical response, okay? All right, so this is the mechanical model can explain tension remodeling and stabilization. And so what underlies this junctional mechanical response is a sort of feedback between tension and strain, okay? So we wanted to know what actually happens at the molecular level. So that's our ongoing work, which is recently came out in preprint. What's really stabilizing the sort of motion of the vertices? Like when you apply a sort of a low light intensity of rho A, you see the junctions contract, but the vertices sort of stabilize, right? So what underlies that? So to do that, what Kate did was to actually activate rho on half junction, okay? And it saw that the junction that you activate do not move as much as the junctions you don't activate, okay? So in a way, rho activity was inhibiting motion, okay? And what we found, I'm going really fast over this, but what we really found is that once you activate rho, there is rapid accumulation of E cadherin, okay? And that sort of limits motion. So there is some sort of positive feedback between rho and E cadherin, okay? So with more rho, you have accumulation of E cadherin, and this force-dependent strengthening of intercellular adhesions can actually limit the motion, right? Okay, so all this necessarily mean that we have quite a complex mechanochemical feedback system, and which is sort of prompting us to reevaluate some of the models that we were confident of, and And so in summary, I just wanted to mention 4 points that I talked about today. So junction remodeling is really useful for collective shape changes and movement, right? And under the hood, what happens is that there is remodeling of tension, which can lead to permanent length changes. And without that, you should never have these intercalations, okay? And also an important feature of epithelial that I found is that there has to be some sort of way to relax the stresses that are imposed onto the system. Like the system must lose some mechanical memory over time, okay? And in recent work, we found that there's also feedback between intercellular adhesions and row activity, right? So, so that's all for today. I'll take questions, and I want to really thank Mike Staden, who did all this work. We moved to Carnegie Mellon last year, and you know, my group is actively looking for postdoc and PhD students who would be interested in doing modeling and theory in close collaboration with experiments, you know, to drive the field forward. So, you know, I'm happy to chat with anybody. And thank you.
Awesome. Thanks, Sheila, so much. All right, we will start with a question from Suvik, asking about, and from the earlier part of your talk, how the aspect ratio of the cells near the wound changes during closing, and does it follow a certain distribution?
Yeah, so that's a great question. I don't have the data here, but if you, yeah, so basically the aspect ratio of the cells don't change because you have intercalations and that sort of relaxes. If you didn't have intercalations, then the aspect ratio gets longer. So in fact, if you go into this paper that we published, we do have the data that's showing that in Drosophila wing disc, where you have intercalations, aspect ratio is maintained over time, but in Drosophila embryo, where you don't have much intercalations, there the aspect ratio increases. Okay. So, yeah.
Thank you so much, Sheila. Our next question is from Raphael. He wants to know, in the elongated wound healing, Raphael is wondering that in the experiment, the speed of the movement at the top point was faster than the lower point, but that was not so evident in the simulation. Is that the right impression? And do you have plots of the speeds of these 2 points as a function of time?
Yeah, I mean, you can just calculate the speed from the slope of this curve. As you can see that in the simulation, indeed, the top moves faster than the bottom, okay? And the reason is geometric. So this has a sort of a shape where you have a high curvature at the top, right? Very pointy, and a lower curvature at the bottom. And if you have purse-string assembly, purse-string forces are higher at regions of high curvature. So that actually drives this faster movement. Yeah, so because they're on different scales, the experimental and the theory data, so that's why they might look like it's not. But yeah, so it captures the features.
Thank you so much,
Okay,
Sheila.
great. Ankita, do you want to do your question next?
You can go with the other question. I can ask Sheila after.
Okay. From Michael Norton, are there any continuum models that are able to capture similar phenomena? If not, what are the fundamental limitations?
Yeah, so I spent a long time doing continuum models and I've lost faith in them. And you know, you can also see in my reviews I wrote a few years ago. But the continuum models, the problem is that you have to pre-assign material properties to the system. So you have to come up with some constitutive laws. And you have to pre-assign, okay, these are the mechanical properties. So there is no way to have emergent mechanical properties in this system. So, I mean, you can construct continuum models that'll be very challenging, but you know, those type of models don't actually at the moment capture this sort of mechanochemical feedback at the scale of individual cells. So I think the cell-based models are useful because you can model a lot of activity that happens at the single cell level that is actually lost in continuum models, okay? And the answer is, yeah, actually depends on, yeah, so those are the fundamental limitations, but continuum models are actually useful if you want to look at macroscopic behaviors, yeah.
Thank you, Sheila. I had a question if you're okay to stick around.
Yes.
I was wondering in the Drosophila embryo, early embryogenesis, there are actually two pools of myosin. There's one on the junction, and then there's apical actomyosin contractility, which is going on. And I know in Rousey's paper, they showed that it's not the junctional myosin and rho activity that is responsible for the junction shrinkage, but rather the apical actomyosin. How does that sort of fit in this kind of a model? And do you have an impression of what that sort of pulsatile myosin does to fluidity of the cells?
Yeah, so here actually we didn't look, that's a great question. We didn't look at two pools of myosin. We were only looking at junctional rho pulses. But if you have medial actomyosin fluctuation, that can also affect the elastic properties of the cells, So, yeah. I mean, so that also plays a role in ratcheting because that actually controls the overall area of the cell. So yeah, I mean, I agree with you. So we need to look at that. But so one way to do that is to use optogenetics over the entire body of the cell rather than at the junctions. Then we can actually study what happens. But
yeah, also I think in the KCO2 cells, I'm not sure if there is any apical pool as such. So that's why I was thinking of these appear as two like independent systems. Because here the pulsatile activity is really on the junction, where there it's actually the apical one. And a junctional myosin is just acting like a stabilizing.
Yeah, I know. So the KCO cells is different because here we're inducing. So I mean, yeah. I don't know. So I think Stefan Arenzes did some optogenetics like that. I mean, yeah, great question.
Okay. We have more questions trickling in, I think. I can take the next question from Mete. Is the physical strength or contraction that triggers ecadherin internalization? So he's wondering if the physical strength or the contraction in the junctions that are triggering ecadherin internalization.
We don't. I didn't talk about ecadherin internalization in my talk. What I was talking about later on is that there is recruitment of ecadherin to tricellular vertices upon contraction. Okay. And this is not about internalization. And yes, this is triggered by, rho A. So what happens is that ecadherin is like a bond. It's like a clutch bond, right? And these bonds are strengthened by forces. That's what is happening here. Okay. I don't know if that answers the question, but that's what I was talking about.
Okay, great. And Mete, if you want to jump in to clarify or ask further, feel free. Our next question from Suvan is noting that at the end of your talk, you said that junctional remodeling promotes collective cell migration. And so with that in mind, does the number of gap junction proteins, for example, connexins, is that proportional to the velocity of cell migration? I guess more broadly thinking about what other kinds of remodeling and do gap junctions, are they contributing to the remodeling at the cell junctions?
I don't know actually. So we haven't done that experiment. Yeah, so usually the number of proteins that are gap proteins would contribute to the adhesion strength. So the adhesion strength should scale with the number of gap junctional proteins. So you need to have them turn over to have junctional remodeling. Whether that correlates with the velocity of cell motion or not, I don't know. I haven't seen the data, so
Yeah, that's interesting because I do think that those of us who look at these kinds of models, we tend to get pretty focused on, cadherins and often ignore thinking about a lot of the other junctions for simplicity.
Yeah.
We can take one more from Jessica, says, great talk. Following up on the observation that inhibiting myosin 2 activity increases tissue fluidity, also inhibits pulse string driven contractility and increases wound closure. Why do you think tissue bothers to generate a bursting in the first place?
So it's essential to generate the initial driving force, right? So you need to sort of kickstart this system. So that's what burstring is good for. So it assembles, it generates huge tension that sort of kickstarts the system, and that forces then, you know, junction, remodel the junction. So yeah, that's what it is, I guess, you know. But it is not only that. So that the field kind of thought before this work with Yanlan that, you know, this tensile actomyosin structures are what is necessary for wound closure, but too much tension is not good, okay?
Thank you so much.
That should be our new life motto. All right, I think that's we can sort of wrap up now, unless anyone wants to jump in with last minute questions for either speaker. But I did want to follow up just before we end things. We had one leftover question for Margarita from YouTube. So Cecilia, who actually gave a talk a year and a half ago, a while back at the seminar series, wanted to know whether the cells undergo apical basal growth after migration.
Yeah, no, thank you, Cecilia. This is also a great question. So it's not something, the mark I'm using now is actin, and I do not see huge cell rearrangements. I don't see huge, actin rearrangements. So at this stage now, I don't see this, but it's definitely something I should look at later stages. Yeah.
Okay, great. And she was on YouTube, so I don't know if she's still here, but hopefully she was able to see that.
Yeah.
All right, thanks, guys. Actually, Sheila, I have one more, just sort of a perspective question for you, if you don't mind. I know we're getting a little bit past the hour. I'm curious your take on how jamming fits into this, and you know, do you think that what we're seeing is really an unjamming transition in the fluid regions versus jammed, or are you looking at fluidization as, you know, something separate than jamming, or are these just terminology and, you know, it's all the same phenomenon?
Yeah, we did not rigorously characterize if it is, you know, jamming, but what's really happening, I think, is local unjamming, not so
local rearrangements being a local unjamming, basically.
Yeah, so it is, I think, a completely unjamming transition, but we haven't done the rigorous analysis because I think that's not very helpful. But you know, the understanding is that there is fluidization and that drives movement. You know, yeah. So that's unjamming.
Great. Yeah. Yeah, I'm always interested, you know, because I think a lot of us are using different words to describe similar phenomenon, or the same words to describe different phenomena, you know, with this sort of new area, I think. So I'm always curious what other folks are thinking about it. Okay, I think with that, we can wrap up. So thank you so much to both our speakers, Margarita and Sheila. Those were really fantastic talks. And we are just going to have Margarita's talk up for the next day or so, and then we'll take it down for now from YouTube. So just so you know, and yeah, all right. Thanks, everybody.
Thanks a lot.
Thank you.
We'll see you next week.
Thank you. Bye.
Bye.